So this is our third chunk of material regarding lipids. This is going to be kind of a shorter lecture because in class we're finishing up an activity, and some of these topics we've even touched on a little bit. But I feel like these are really important for understanding understanding membranes and lipid mobility and membrane composition of lipids. So if we look at here, we've got a blue headed lipid. So there's our phospholipid or our glycerophospholipid as an example. And that molecule is moving to the other side of a membrane. So it's going from like the inner leaflet to the outer leaflet, or the outer leaflet to the inner leaflet. And I want you to be familiar with and comfortable with that terminology. Outer leaflet, inner leaflet. So if you think about, most plasma membranes, your inner leaflet is the cytosolic side, and your outer leaflet would be the basically the side outside of the cell. That movement is going to happen very slowly, although it can happen. Contrary to that is our lipids that are moving laterally. That's going to happen very quickly. I always kind of think about it as or for some reason I think about a membrane as being similar to basically like a wave pool where people are always moving, and if you move from one place in a wave pool to the next, it just takes a little bit of time. Now, the fluidity of membranes is going to be influenced by temperature. So you have a transition temperature, or every membrane is going to have a transition temperature, and whenever you are below that transition temperature in a cooler environment, your structure is a little bit more rigid, more I don't know if I would say structurally sound, but rigid definitely. There's more order to it. Whereas when you're above that temperature, then we have something that's a bit more fluid and really a bit more chaotic. So image A is showing our gel-like paracrystalline structure or paracrystalline state, so the more rigid version of our lipid, and then B is showing our fluid at above the transition temperature. Now, the transition temperature is dependent on chain length and the degree of saturation of its component fatty acids. So cholesterol is going to broaden the temperature range of the phase transition. Now, this is a figure that I think is very important to understand, and very important to understand what's taking place here. So this has a little bit less to do with the transition temperature and more to do with basically a membrane maintaining the same or a uniform rigidity, whether it's grown at 10 degrees or 40 degrees Celsius. And I want to draw your attention to these two lipids at the top and then these two down here. At a low temperature, we have a high percentage of monounsaturated fatty acids. At a high temperature, by contrast, we have a high percentage of saturated fatty acids and a low percentage, by comparison, of your unsaturated fatty acids. So going from 10 to 40 degrees Celsius, we see a drop off in the number of fatty acids that are unsaturated, where we see an increase in the percentage of saturated fatty acids. Now, what makes up a lipid membrane? Remember, we've got the inner leaflet and the outer leaflet. The total phospholipid, we've got an even distribution, 50-50, 50% on the outer, 50% on the interior. Now, sphingomyelins, the majority I apologize. The majority of your sphingomyelins are going to be found on the outer side of the leaflet, which, if we think back to those sphingomyelins, those were something like our gangliosides or ceramides. Those were our means of communication. Phosphatidylcholine is typically found on the outer leaflet, phosphatidylethanolamine typically on the inner, and phosphatidylserine the same thing. Now, keep in mind that the lipids that are found on the inner leaflet can flip over and be on the outer leaflet. And there are certain circumstances that are going to stimulate that. So movement from the inner to the outer leaflet, just to kind of signal, hey, this is what's going on in the cell, might take place. If I remember correctly, phosphatidylserine typically flips to the outer leaflet in mammalian cells in autophagy, or whenever cellular senescence begins, also known as autophagy. So whenever the cell begins to basically recycle all of its pieces, then you'll see something like phosphatidylserine flip to the outside, so that then macrophages can be attracted, bind onto that phosphatidylserine, and munch up whatever they're bound to. Now, phospholipids are synthesized on or near the interior of the membrane. There are two mechanisms to get the phospholipids from one side of the membrane to the other. Flipases are enzymes that catalyze the flip-flops of specific phospholipids in what is known as a facilitated diffusion. So, like I said, the movement from the outer to the inner, or the inner to the outer, that's going to happen very slowly, but awesomely there's a series of enzymes known as flipases that will make that action happen very quickly. Now, flipases tend to equilibrate the distribution of their corresponding phospholipids across a bilayer. Phospholipid translocases transport specific phospholipids across a bilayer in a process that is driven by the hydrolysis of ATP. Translocases can establish a non-equilibrium distribution of phospholipids. Membrane composition. Biological membranes contain proteins and lipids. Proteins catalyze reactions, mediate flux of nutrients and waste, and participate in relaying external information to the cell. Protein to lipid ratios vary with membrane function. So if we look at these different cell types, a human myelin sheath cell, 30% protein, 30% phospholipid, so that's 60, and then 19% is sterol, so about would be 79%, or nearly 80% of the lipid, or of the membrane's weight, is attributed to proteins, phospholipids, and sterol. Now, if we compare that to something like yeast or E. coli, well, yeast and E. coli both have a very high percentage by comparison of their membrane attributed to proteins. Phospholipids for yeast are about 7%. E. coli is about 25%. So 100% of the weight of an E. coli membrane is attributed to basically those two things, proteins and phospholipids, and 0% to sterols. Why? Because E. coli don't make sterols. So easy explanation there. Now, with that, sorry. It's going to vary a little bit based on the type of cell as well. So that's a human myelin sheath cell. Endothelial cells are going to have a different composition. But those are the major components. Your proteins, phospholipids, and steroids are going to be the major weight-bearing components. So membrane structure and assembly. The phospholipid bilayer is a fluid matrix. The bilayer is two-dimensional solvent. And here's essentially what it looks like. This is pretty overwhelming. There's a bunch of stuff here. So I'm going to finish out the bullet points here. Lipids and proteins can undergo rotational and lateral movements, so they can go this way or they can go like this. Membranes are asymmetric, and I believe that's the last item on this slide, so I'm not going to press next because otherwise it would progress my slides. Now, anytime you look at any sort of depiction of a lipid bilayer or a membrane, I think that it's always important to remember, well, which is the inside and which is the outside. Where's my inner leaflet? Where's my outer leaflet? Well, here we've got inside. This is my cytosolic side. This is outside. This is my extracellular side. Now, what I want you to notice, and what's absolutely important to recognize, is that when you look at glycolipids, oligosaccharide chains of glycoproteins, those are displayed on the outside. Those, if you remember when we talked about gangliosides and sphingolipids and sphingomyelins, those are communication mechanisms. Specifically, if you look at a glycolipid here, that would be a good example of a sphingolipid. That would be a good example of a ganglioside. So if you look specifically at that, we've got these rings, 1, 2, 3, 4, these 5 rings, 5 sugar rings. One of them is sialic acid when we're talking about gangliosides. Those are our mechanisms for communication. So you want to be able to communicate outward, because a cell is kind of like a person. Nobody can read your mind. So without displaying what's going on inside, the other cells aren't going to have any idea of what's going on. So when a glycolipid is displayed outward, that gives another cell a means to say, oh, okay, I see that you're this type of cell. Therefore, I don't interact with you. Now, with that in mind, those are things that are displayed outward. What I would also like to draw your attention to is, well, what's on the inside? And there's a bunch of stuff that's labeled on the inside. So let me erase all my markings for outside components. Now inside. So first of all, what's pointed out on the inside, I'm going to go from the left to the right, and I'm going to start kind of top, and as I have to go down, I will. Phospholipid polar heads. So those are our glycerol backbones with a phosphate group, and there might be a head group there. There might not be. Those are displayed on both the interior and exterior of a cell. Those blue circles, those represent our phospholipid polar head groups. We also have a peripheral protein here. Now, what's important whenever you look at this peripheral protein is it's got a bunch of coils, and some of these coils get in close contact to those phospholipid polar heads. Notice how this protein, this polypeptide chain, is not embedded, it's not inserted into the lipid bilayer. So a peripheral protein is peripherally, or it's on the periphery of a lipid bilayer. Now, in comparison to that, if we jump over here, an integral protein, that means that protein has been integrated within the lipid bilayer. This is a single transmembrane helix, and this integral protein's got one termini on the inside of the cell and one termini on the outside of the cell. Next up, we have a peripheral protein covalently linked to a lipid. Okay, so this contact is more than likely going to be a lipid has been added to the protein, so post-translational modification, adding something like palmitic acid to that protein, so that that lipid is embedded into the membrane, and that protein by association is embedded into that membrane. That's different from our peripheral protein over here, which really is not embedded into the membrane. It's just kind of tangentially, it's in contact with. So think about this peripheral protein being R groups interacting with polar head groups, whereas our covalently linked lipid, that is a lipid that's embedded in the membrane that has been added to an R group of our protein. So this is not necessarily an interaction with a polar head group. Now next up is our integral protein with multiple transmembrane helices. Now if you look at this, start at this end over here, and you kind of follow it, goes up and out, and then it loops back, and there's another transmembrane helix, and then it goes up and out and back, and here we go. So this is a protein where this termini and this termini are on the exact same polypeptide. Now it's a very long polypeptide that gives us approximately 4 transmembrane domains, but this is a protein that loops up, out, up, out. It's kind of like a very long shoestring there. So we have proteins that have light contacts, that's our peripheral protein, so R groups plus head groups interact. We have integrated proteins with a single transmembrane domain. We have lipid-linked proteins. We also have integrated proteins with multiple transmembrane domains. Now we're going to dissect these a little bit more in the next slide, but for the time being, that's what I need you to know about all of them. So here's another depiction of this, and it's giving us a little bit more detail. There's not a lot of information that's different. The one thing that I didn't really point out on the previous slide, and that wasn't like intentionally, was cholesterol. cholesterol is another component of our membrane, which we saw whenever we were looking at the percentages. But cholesterol here is depicted as kind of little, I don't know, little droplets. So these little molecules here. Cholesterol is a part of a membrane. Cholesterol is going to modulate the fluidity of a membrane. And so the more cholesterol, the more rigid that membrane is going to be, the smaller amount of cholesterol, the more it's going to be, its flexibility is going to be dictated by lipid composition. All right, now integral proteins are associated tightly and require detergents or chaotropic agents to separate from the membrane. So if you want to extract that protein, well then that's what you would have to do. You'd have to use something like a detergent or a compound known as a chaotropic agent. Lipid-linked proteins tend to be globular after their release. So when you think about the hydrophobicity of these proteins, a lipid-linked protein isn't necessarily hydrophobic or doesn't have a long stretch of amino acids that are hydrophobic, whereas an integral protein is going to have a region that is very hydrophobic because the interior of a lipid bilayer is going to be hydrophobic. Peripheral proteins are globular-like and are easily dissociated from membranes. Now here's a way that we look at our different types of transmembrane proteins. These are all transmembrane proteins with helical regions. These helical transmembrane proteins, they fall into a couple of different categories, and they vary based on, well, their association with a membrane and kind of how they fall. So one thing that I want you to notice, and what I think is really helpful, is to think about these all as very similar proteins. They're transmembrane proteins. We look at type 1 and type 2 transmembrane proteins. What do they have in common? Well, they have a single transmembrane alpha helical domain. So this is an alpha helix, so that's the secondary structure that's formed, and that is what's embedded in your membrane. They've got an N termini on the outside and a C termini on the inside. So inner leaflet is where you would find the C, or you'd find the C termini closest to the inner leaflet and the N termini closest to the outer leaflet. Now if we go on to look at type 2 transmembrane proteins, the N termini of the protein is inside the cell, or inside, nearest the inner leaflet, whereas the C termini is on the outside. Okay, so type 1, type 2, they're basically just inversions of one another. Now type 3 and type 4, these two proteins are going to differ, and the way that they differ is type 3s are, here's a termini, here's another termini. Type 3s are one entire polypeptide, whereas type 4, let's look at these. There's an N termini, or there's a termini, here's a termini, here's a termini, here's a termini. So when you have those multiple termini like that with a type 4 protein, that's multiple polypeptides. Now these multiple polypeptides, I always kind of look at them as like, you know, let's say that this marker here is one of these proteins. Here's another. Well, these kind of aggregate together and they form a structure embedded within the membrane. Here's a third one. Okay, and that structure, when you look at it from the top down, that's what it looks like. I like to think about these type 3 or type 4 for that matter, because it's a bunch of alpha helices, but what they can do is they can open up and make pores, make regions within the membrane where nutrients can flow through. Okay, so type 1 and type 2 are single helices. Type 3 and type 4 are multiple helices. Is it multiple polypeptides or multiple polypeptides, multiple helices is a type 4. One helix, one peptide is a type 3. Now type 5 and type 6, what makes these two different from one another is a type 6 has an alpha helix, but what it also has is, I'm circling it down here, and that's a lipid linkage. Type 5, that is a protein that only has a lipid linkage. So these two proteins are anchored into lipids, are anchored into the membrane with the help of a lipid, but type 6s also have a alpha helix that anchors them to the membrane. So our differences between type 1 and type 2 is the C termini of those proteins or the N termini. Where is it? Type 1, the C termini is on the inside and the N termini is on the outside. Now here's a good depiction of what some of those transmembrane proteins can look like if rather than alpha helices, they're beta sheets. Now, what I want you to look at is just kind of do a quick glance at these proteins and then look at the top-down view. Because effectively all of these proteins have a similar top-down view structure because they're all capable of basically making a little pore in the membrane so that nutrients or molecules can flow through. And they make that kind of opening, that cavity in the membrane, so that, well, they can selectively let things through. Good examples of this would be like, what are they? Well, your sodium-potassium pumps have similar structures to this, but also your, I think it's the nicotine receptors, effectively. What they're able to do is they open up and they let a bunch of calcium through. Now, one thing that we alluded to a little bit ago was our lipid-linked proteins, our type 5 and type 6 proteins. They have lipid linkages, and you might think, well, what lipid? Well, these lipid linkages are derived from the same molecules that are used for synthesis of cholesterol. And those are those 5 carbon long isoprenoids. And they make structures like this. Farnesyl right here is a molecule that can get anchored to a protein. So just imagine I'm writing pro as best I can with my mouse. Protein right here, and yeah, there's our protein. And this molecule there's 5 carbons, here's 5 more, and then here's 5 more. This is a 15 carbon long molecule that gets anchored to a protein. Now what I want you to notice about this 15 carbon long molecule, or what I want you to think about this 15 carbon long molecule, is 15 carbons. Sheesh. Carbons and hydrogens. Have we seen chains of carbons and hydrogens before? We absolutely have. Our fatty acids. Now, when we think about a fatty acid, we've got a molecule that's amphiphilic, so something that has a polar and a nonpolar region. Does this look like it has a nonpolar region? Totally. That 15 carbon long chain, super hydrophobic. So that's going to love to associate with the interior region of a lipid bilayer. Same thing for geranyl residues. This is 20 carbons long. Now, my point is that these are where proteins can be modified and get something like a geranyl residue or a farnesyl residue. And these anchors are going to kind of insert themselves into a lipid bilayer, and they're going to be kind of stuck there. And that's going to mean that protein's going to be stuck there. Now, with all that in mind, we've also got these anchors over here. Now, this is a pretty big anchor and a pretty big structure. What I want you to do when you're looking at this, though, is look at this right here. That's 3 carbons. Those 3 carbons make up a glycerol molecule, effectively. And those 3 carbons have, well, there's a fatty acid. Lo and behold, there's another fatty acid. Here's a phosphate, a glycerophospho. Oh, and there's an inositol head group. So now we've got a phosphatidyl inositol, which is written down there. And from that phosphatidyl inositol, there can be a 4 sugar long link that then connects a phosphoethanolamine group to a protein. So when you see a phosphatidyl inositol molecule, I'm going to write mem. This is my membrane right here. This that I'm putting a big box around and circling, this is inserted into the membrane. Then we have these long links that connect a protein to a molecule that's connected to a membrane or anchored in a membrane. This is known as a or a phosphatidylinositol linkage to link it to embed a protein in a membrane, or a GPI linkage. Now, with that in mind, what we were just talking about is basically this down here. Here's our glycerol, here's our phosphate, here's our inositol head group, here's our sugar linkage, here's our phosphatidylinositol, and here's our protein. Okay, so that's what we were just looking at. These other groups up here are ways that we can link things like farnesyl or a geranyl group to a protein. Now, I want to go into each one of these because it's not just farnesyl or geranyl that links a protein and gets it associated with a membrane. You can also have palmitic acid, which is a fatty acid. Palmitic acid can be added to a cysteine or a serine residue. And whenever that's linked to a cysteine or a serine residue, it's a thioester-linked fatty acyl anchor. Amide-linked myristyl anchors. Myristic acid is another fatty acid. So if I remember correctly, palmitic acid is, I think, 18 carbons long, and myristic acid, I think, is 16 carbons long. So these fatty acyl anchors are going to kind of be hooked up to a protein and just basically stabbed into a membrane, and they stick around there. Thioether-linked prenyl anchors. Remember, our 5-carbon long molecules are isoprenoids, also known as isoprenes or isoprene units. These 15-carbon farnesyl and 20-carbon long geranyl residues are typically added onto cysteine residues. Let me go back for a second because our myristic acid is going to be added to a glycine. I want you to know these amino acids and where these different groups are added. So palmitic acid can be added to a cysteine or a serine. Myristic acid can be added to a glycine. A isoprene unit can be added to a cysteine. And then last but not least, our GPI linkages, those are going to be added to a C-terminal amino acid. And it can vary which amino acid it's linked to, but it's going to be on the C termini of the protein.